Well, thank you very much for that great introduction today and for hosting this webinar for us today. Good morning again, and good afternoon, good evening to all of you out there, depending on where you are in the world today. Thanks again for taking the time to join us. And I'm very happy to present this webinar today on non-CYP mediated metabolism pathways and in vitro evaluation strategies to assess these non-CYP mediated metabolism pathways. So just to give you a brief outline of what I plan to present today, we'll first start by talking about are CYPs being minimized in drug design. We'll then talk about choosing the appropriate in vitro test system for non-CYP metabolism studies, and then look at some of the basic strategies that can be employed to assess what enzymes are involved in the metabolism of the drug. We'll follow up looking at why looking at non-CYP metabolism is important, looking at a couple different case studies, and then we'll have some time for questions from you at the end. Now, just to start out, I'd like to provide a list of abbreviations. Now, I'm not going to spend any time to go through this alphabet soup of abbreviations listed here, but I'm just putting it here for reference, so you can go back and look at these later. Okay, to start out, is the role of CYPs or CYP metabolizing enzymes being minimized in drug development? So medicinal chemistry works sometimes in minimizing the role of cytochrome P450 enzymes, more commonly known as CYP enzymes, involved in the metabolism of xenobiotics. So one major reason for this is the desire to decrease the drug-drug interaction potential of a drug. Additionally, reducing the drug's liability to be metabolized by CYP enzymes could be employed to either lengthen the drug's rapid metabolism or to shorten the drug's longer metabolism. And again, this is just employed depending on the desired effect of the drug in question. So the reference highlighted here at the bottom of the slide, this information was presented by Ph.D. and Dalvi in Drug Metabolism and Disposition in 2016. And this was a special section related to non-CYP metabolism. So I've highlighted here some of the commentary by the authors of modifications that can be made to molecules to shift their metabolism towards different enzymatic groups as opposed to cytochrome P450 enzymes. For example, to shift a molecule more towards metabolism by UGT or sulfonyluretransferase enzymes, nucleophilic groups such as aliphatic and aromatic hydroxyls, carboxylic acids, amines, and hydroxyamines, thiols, can be added to the molecule. To shift more towards aldehyde oxidase and xanthine oxidoreductase, heterocycles with low electron density can be employed. To shift towards more towards flavin monooxygenase, soft nucleophiles can be added, such as sulfur or nitrogen heteroatoms. To push towards carboxyl esterase, ester moieties can be added, and this is often a tactic that's employed for prodrugs. Carbonyl reductase and aldehyde reductase, to shift more towards metabolism by these enzymes, ketone and quinine moieties can be added. Monoamine oxidase enzyme metabolism can be selected for by adding amines. And lastly, other hydrolase enzymes can often be exploited for prodrug activation. And again, this can be selected for by adding esters or amide moieties to the molecule. So in spite of all the work in medicinal chemistry, the reports of the decline of the role of cytochrome P450 enzyme involvement in xenobiotic metabolism might be a little premature. So the data on this slide were put forth by Cerny in the same 2006 edition of Drug Metabolism and Disposition that I mentioned earlier. So if we take a look at these plots, Cerny looked at the drugs that have been approved between 2006 and 2015. And you can see here he broke them down into three different groups, one from 2006 to 2010, 2011 to 2015, and then the data set as a whole from 2006 to 2015. And if we look between 2006 and 2010, about 43% of the drugs that are approved were metabolized by CYP enzymes, with about 29% of the drugs being metabolized by non-P450 enzymes. Between 2011 and 2015, the amount of drugs approved that are metabolized by CYP enzymes increased to 58%, and non-CYP enzymes metabolizing about 30% of the drugs that were approved. And when we look at the data as a whole, about 53% of the drugs approved are metabolized by CYP enzymes, with about 30% being metabolized by non-CYP enzymes. Now, I would agree with Cerny that while, and he quotes, significant, the relative contribution of non-P450 enzymes to drug metabolism does not appear to have increased dramatically over the last 10 years. And drugs metabolized by CYPs seem to be unlikely to be of little concern anytime soon. Now, most of the reaction phenotyping studies that we conduct at CYNOTech, of course, tend to be focused on P450 enzymes. And the second major enzyme that is the focus of reaction phenotyping studies that we get requests for would be for the UGT enzymes. Now, we do get requests for more special cases of different enzyme families to investigate, but those are more rare. So let's take a look at some of this data a little bit more in depth. Now, if you look at this graph, this is the same data presented by Cerny. And if you look at this graph here, he excluded prodrugs from this analysis. So if we look, there was a total of 125 drugs that were approved that were not prodrugs between 2006 and 2015. And we could see that about 53% of these drugs that were approved were metabolized by P450 enzymes. This was followed with the second highest being UGT enzymes, and about equal would be other hydrolase enzymes. Rounding out this list would be carbonyl reductase, aldehyde oxidase, and other non-CYP metabolizing enzymes. Now, I'd also like to present some data that was put out by Sarvana, Kamur, and colleagues in 2019. In this paper, they were looking at 250 drugs that were approved between 2005 and 2016, again, excluding prodrugs in this analysis. But they did look at the metabolism of the active moiety of these drugs. So similar to Cerny's findings, they found that out of all the drugs that were approved, a majority of them were metabolized by UGT enzymes. And again, this is looking at the major non-CYP enzymes. So other than CYPs, a majority of the enzymes involved in the metabolism of these drugs were UGT enzymes. If we look at the rank order of these non-CYP enzymes, we see again that UGTs make up about 38%, followed by other enzymes making up 30%. Carboxylesterases were found to contribute to about 10%, followed closely by esterases at 9%. FMOs or MAOs contributed about 6% to the total. And rounding out the list are the sulfonyluretransferases, AO or XO, and aldehyde dehydrogenase or alcohol dehydrogenase, following out the list at about 1%. And similar to Cerny's finding, the percentage of drugs that were metabolized by CYP enzymes made up about 54% of the total. Now, another way to take a look at these data is to rank the top 200 most prescribed drugs. Now, again, this analysis is excluding prodrugs and is also looking at the metabolism of the active moiety of the top 200 prescribed drugs. So as you can see, again, CYP metabolism makes up about 50% of the total. And UGTs are by far the majority of the major non-CYP enzymes involved in metabolism. Now, the rank order is slightly different when we look at this based on the top new approved drugs, but the outcome is basically the same. So as you can see here, for non-CYP enzyme groups, UGTs contribute about 45% for the metabolism of the top 200 prescribed drugs, with other enzymes making up 14%. Esterases make up about 12% of the metabolism of these drugs. In this case, when we look at the top 200 prescribed drugs, sulfonyluretransferases contribute to about 10% of the metabolism of these drugs. Alcohol dehydrogenase, an aldehyde dehydrogenase, make up 7%, as do carboxyl esterases. And rounding out the list would be AO and XO enzymes, followed by FMOs and MAOs. Now again, CYP enzymes are still responsible for close to 50% of the metabolism of the top 200 prescribed drugs, and in this case, making up about 48%. Now the other enzymes category includes biotransformation reactions, such as oxidative dealkylation, acetylation, deiodinase, deethylation, pyrimidine catabolism, and dehydropeptidase and thiamine phosphorylase enzymes. Now, what about prodrugs? Since the data sets that I was just sharing with you excluded prodrugs, both authors actually looked at prodrugs in their own category. So in Cerny's analysis of FDA-approved prodrugs between 2006 and 2015, he had a data set of 22 drugs that were approved. Now, out of these prodrugs, 86% of them were metabolized by hydrolases. This was followed up by equal proportions by kinases, glutathione, and other forms of chemical activation of the drug. Sarvana Kamur, in their analysis, looked at the established prodrugs between 2005 and 2016. And this data set had an N of 9. So the activation of these prodrugs, again, was predominantly driven by hydrolase enzymes that made up about 78% of the enzymes involved in the metabolism of these drugs. And this was followed by demethylases, making up 22%. Now, for FDA-approved prodrugs between 2005 and 2016, was a larger data set in this case, had an N of 13. And again, hydrolases made up a majority of the enzymes involved in the metabolism and activation of these drugs, being 75%. In this case, the second most prevalent non-CYP enzyme were the kinases, making up 25%. So I really briefly want to go into some of the caveats between these two papers, because it's important to understand this when looking at what are the most important non-CYP enzymes. So the first of these are the limitations in the data available. As you know, all the data is not always available. You just may not have the full metabolic ID nor mass balance for a given drug. And this information might be incomplete for some of the drugs. And I'm sure that anyone that has reviewed the investigational brochures or FDA, NDA briefing documents for some drugs realize that there's gaps. It's just part of the programs for certain drugs. Second, glucuronides are generally unstable in feces and sometimes in urine. And I don't think it's always completely appreciated that they are unstable. So their complete contribution to the overall metabolism of a compound may be underrepresented in these analyses. Third, multiple step oxidations can involve both CYP and non-CYP enzymes at the same time. And also, multiple minor metabolites may be formed by the same enzyme or enzyme group. So while individually each enzyme may be minor, collectively as a given group, they may make a large contribution. And in this case, they would also be underrepresented in this survey. Contributions of one enzyme over another may be over or underestimated because no corrections for fraction metabolized were applied. So either the fraction metabolized wasn't required for the study, or the data were not available for some other reason. And lastly, both publications lack data from 2017 to currently here in 2022. So drugs from 2005 to 2016 may represent drugs that were in discovery 10 to 20 years prior. So this is a look backwards. And it could be in 5 years from now, we'll be saying that other drug metabolizing enzymes are more important than we're saying that they are now. But again, I don't think this is happening anytime soon, but it's definitely something to keep in mind. So based on these two papers and everything I just said, and some of the studies that we see here at Sekisui XenoTech, I've just made a quick list of major human non-CYP enzymes involved in small molecule drug metabolism. This is kind of like my top 9 list. Of course, first would be the UGTs, followed by the hydrolases. And these can be broken up into the numerous hydrolases that are out there. I'm separating out carboxylesterases from some of the other hydrolases. Carbonyl reductases, followed by FMOs and MAOs, sulfuryltransferases enzymes, aldehyde oxidase or xanthine oxidase, alcohol dehydrogenase and aldehyde dehydrogenase enzymes, and lastly, the other. These include cytidine, nucleotides, GSTs, other enzymes that are involved in oxidative dealkylation, dehalogenation, pyrimidine catabolism, etc. So I'd like to just briefly talk about how the regulatory agencies address non-CYP metabolism and how the guidance approaches dealing with enzymes that are non-CYP based. So I should point out that the FDA does mention carboxylesterases, which the other 2 guidance documents do not mention, that being the European EMA and Japan's PMDA. I should also point out that Japan's PMDA does highlight DPD, dihydropyrimidine dehydrogenase. Now, dihydropyrimidine dehydrogenase is not specifically named by the PMDA. However, it's implied by reference to the fatal interactions between cerevidine and 5-fluorouracil, which were famous in Japan for quite some time. Now, the EMA does mention UGTs and salts broadly, and also mentions glutathione as transferases specifically. But it then talks about using either hepatocytes or subcellular fractions to be able to elucidate these metabolic pathways. Now, the FDA guidance in its most current form does say, and this I quote, the contribution of a specific metabolizing enzyme to an investigational drug's clearance is considered significant if the enzyme is responsible for greater than 25% of the drug's elimination. Now, this is a pretty clear blanket statement by the FDA that's basically saying, even though we're not specifically mentioning all the non-CYP enzymes that might be responsible, if those enzymes happen to be responsible for greater than 25% of the drug's elimination, we probably want you to take a look and figure out what those enzymes are. I would also like to point out that some of the enzymes that are not mentioned but fell into the top 10, such as all the carbonyl reductases, aldoketo reductases, most hydrolases, although the FDA does mention carboxylesterase. For the other enzyme category, only the PMDA mentions dihydropyrimidine dehydrogenase, and only the EMA mentions glutathione S transferase. Okay, now moving on. I'd like to talk about choosing the appropriate in vitro test systems for non-CYP studies. So it's of great importance to choose the appropriate in vitro test system, especially when looking at non-CYP metabolism. The choice of test system is a very important first step, and this can have implications on the interpretation of the results. And I'll go into this and we'll talk about it with some examples and case studies further on in the presentation. But some thought and prior knowledge is good to have ahead of time. As hepatocytes, which are often considered to be the gold standard of test system for metabolism studies, hepatocytes might not always be the best test system of choice when approaching non-CYP metabolism. So when we rank these various test systems by their metabolic versatility, as shown here, hepatocytes, as I said, are the gold standard. Hepatocytes do have the potential to confound results in some certain cases, however. For example, in hepatocytes, you can have futile cycling, where you have an oxidation and then a reduction back to the parent of the drug. And this cycling can just go back and forth. So you're really not seeing the metabolites that are involved. In hepatocytes, you can also have low levels of metabolites that just aren't able to be detected. And also you have the fact that hepatocytes are just more complicated than subcellular fractions. Not only do you have to choose between suspended versus plated versus cryopreserved versus fresh, there are more choices that need to be made up and decided upon up front when designing the experiment. So when it comes to versatility of the test systems, hepatocytes, of course, are the most versatile since they're living cells with all their metabolic pathways present and in operation, and no need to add any additional cofactors as they're present in the system. The next test systems we have are subcellular fractions. Now, S9 would be the most complete test system after hepatocytes, and that's because S9 consists of both microsomes and cytosol. Now, since S9 is not an intact living cell, in order for some of these enzymes to function, additional cofactors need to be added. And by this, you can control the activity of these cofactor-dependent enzymes. However, in S9, compared to the next test system, the enzymes can be relatively dilute because it's both cytosol and microsomes. The next versatile test system in this ranked list would be microsomes. Now, microsomes often yields metabolites at the highest abundance because they are highly concentrated preparations of various enzymes. Again, since microsomes are not intact living cells, many of the enzymes require the addition of certain cofactors to be added for their function. And this can be a benefit, again, because you can control the activity of these cofactor-dependent enzymes. For example, the addition of UDP-GA for the UGT enzymes. Now, commercially available preparations of pretty much most microsomes are contaminated with outer mitochondrial membrane. So it's important to keep in mind that even in microsomes, there may be some MAO and other mitochondrial enzymes in the preparation that may be active. Lastly, commercially available recombinant human forms of many non-CYP enzymes may also be an option. Now, I highlight the words active because there are forms of recombinant enzyme available that are not active. So it's just something that needs to be kept in mind when selecting and using recombinant enzymes to examine metabolic pathways. So as I've already mentioned several times, with a lot of these enzymes, especially when they're used in subcellular test systems, you can control the activity of cofactor-dependent enzymes by addition or withholding of the appropriate cofactors. For example, NADPH is the cofactor that's required by CYP and FMO enzymes in microsomes. UDP-GA cofactor is required for the activation of UGT enzymes in microsomes. PAPS is the required cofactor for sulfur transferase enzymes when used in cytosol. Acetyl-CoA is the cofactor required for acetyl-CoA synthetase in microsomes, and also for NAT enzymes in mitochondria and cytosol. SAMe is the required cofactor for methyltransferases in cytosol and microsomes. And lastly, some enzymes might require no cofactors or water or oxygen, which would already be present, of course, in the preparation. For example, esterases in microsomes and cytosol, epoxide hydrolases in microsomes and cytosol, and MAOs in mitochondria, and human aldehyde oxidase in cytosol. So typically, when a client comes and has questions on what is the appropriate test system to choose, one of the first things we like to ask for is, what is the structure of the molecule? Looking at the structure of the molecule can provide a lot of information and help assist in the selection of what test system might be the most appropriate one to use. Now, I'm not going to go through this whole table, but I'd like to point out that for some of these functional groups, such as alkenes, aliphatic alcohols, aliphatic amines, while they can all be metabolized by cytochrome P450 enzymes, however, they're mainly metabolized by these other non-CYP enzymes, such as UGTs, salts, FMOs, etc. And in these cases, it's kind of a judgment call as to which test system would be the most appropriate. So you can discern what enzymes might be most active in the metabolism of the drug by running it first in microsomes with and without various cofactors, and then repeating the experiment in other test systems, such as cytosol and mitochondria, etc., both with and without the appropriate cofactors for the putative enzymes that might be involved in the metabolism of the particular molecule. And looking a little further, you can see that all of the functional groups can be metabolized by cytochrome P450 enzymes, but also by other possible non-CYP enzymes as well. And I should note that in some of these different enzymes, or that some of these different enzymes are present at higher levels in extrahepatic tissue. So for example, some esterases may be in plasma, and some esterases may only be found in blood. So again, it can be important to distinguish between these two and maybe run these tests in both or multiple test systems. So as I mentioned, recombinant enzymes are a really important tool to be utilized when elucidating the potential metabolism or the metabolic pathways of the compound in question. So this is just a list of some of the examples of commercially available recombinant human non-CYP enzymes. These are just two companies that I'm listing, Cypex and Corning. And full disclosure, I should just mention that Sekisui XenoTech is a distributor for Cypex. I'm sure that there are other companies that offer other recombinant enzymes out there, but these are just the two big players and have the largest portfolio available. So as you can see, we have aldehyde dehydrogenase 1A1, AO, the carboxyl esterases 1 and 2, various forms of salt enzymes and UGT enzymes, MAOs, FMOs, NATs, are all available out there to be used. And it's important to remember, though, when using recombinant enzymes, that recombinant enzymes provide a largely qualitative results. So just to say that another way, using recombinant enzymes are just telling you which enzymes can be involved in the metabolism of the drug. Now, that doesn't necessarily mean that it would be that they're always involved in the metabolism of the drug in vivo. And lastly, when looking at recombinant enzymes, as I mentioned before, always ensure activity with appropriate positive control substrate. And selective inhibitors might not always be available for all of these various enzymes. And we'll touch on the use of selective inhibitors a little further on in the presentation. Now, I'd like to just talk about and present a basic strategy that can be employed to assess what enzymes are involved in the metabolism of a drug. So these next few slides are from a 2016 IQ Consortium paper that pretty much provides a flowchart to follow when assessing the potential enzymes involved in the metabolism of a drug. So the first question that's asked is the metabolism of the drug predicted to be a major route of clearance? And if the answer to that is no, well, then there's a very low risk of that drug being a metabolic victim or involved in a metabolic drug-drug interaction with other drugs. However, if metabolism is predicted to be a major route of clearance from the drug, we can split that then into two questions to ask next. First, is oxidative metabolism of the compound NADPH dependent? If it is, then we'll test for CYP and FMO mediated oxidation and phenotype these accordingly. If oxidative metabolism is occurring, but it's not NADPH dependent, we'll test for AO, XO, and MAO mediated oxidation, and again, phenotype accordingly. Now, the other question to ask is the metabolism of the compound due to direct conjugation. So phase 2 metabolism, either via UGTs, salts, NAT, or GST. If it is, then we'll test for these various enzyme types and phenotype them accordingly. And if the metabolism is not by direct conjugation, we'll test other pathways, such as metabolism by carboxylesterases or amidase metabolism. So going on to scheme A, if the drug is cleared through metabolism and that metabolism is confirmed to be NADPH dependent, there's two questions to ask. First, does the compound undergo metabolism in recombinant human CYPs? And is the clearance of this compound in liver microsomes or hepatocytes substantially inhibited by ABT? And ABT is a pan-CYP inhibitor. So if the compound undergoes metabolism and its clearance is inhibited by ABT, then yes, we'll phenotype for the various CYPs. And if not, then CYP reaction phenotyping is not required. The second major question to ask is, does the compound undergo metabolism in recombinant human FMOs? And is the clearance of this compound in liver microsomes substantially inactivated by a brief heat treatment in the absence of NADPH? The answer is yes. Then we can estimate the contribution of metabolism by FMOs. The answer is no. Further assessment of FMO enzyme involvement is not warranted. And lastly, the scheme B. So if the drug is cleared through metabolism, but it is NADPH independent, but still oxidative metabolism. Ask the question, is clearance of the compound in cytosol inhibited by hydrolysine? If the answer is yes, we'd assess the contribution of its metabolism to aldehyde oxidase. If no, then we look at the clearance of the compound in cytosol and see if it's substantially inhibited by allopurinol. The answer is yes. It points to the involvement of exoenzymes, and we can further investigate that. And if the answer is no, then AO and exoenzymes are not involved. Lastly, does the compound possess primary, secondary, or tertiary amines, and is it metabolized in recombinant human MAO and in the mitochondrial fraction? The answer is yes, and the clearance in the mitochondrial fraction is substantially inhibited by MAO inhibitors. Then we'll assess the contribution of MAO enzymes. And if it's not, no further assessment of MAO metabolism is needed. Now, I should point out that this is just a general guidelines and a basic strategy to be followed. It does not need to be followed specifically in this order. Depending on where one would start at the reaction phenotyping and determining the metabolic pathway of their drug, as I already mentioned, can depend on several factors, such as the structure of the drug or any other information that the drug developer might have at hand. Okay, now I'd like to just move on to why looking at non-CYP mediated metabolism is important. And we'll look at this in the context of UGT enzymes, showing a couple of case studies, and then show some strategies, again, that can be employed to look at non-CYP metabolism. So the first example here is a glucuronide metabolism case study example. So the compound gimfibrazole is metabolized to an acylglucuronide that can cause irreversible time-dependent inhibition of CYP2C8. So these are two inhibition plots shown here. These plots are the same except for the drug that's being tested. On the x-axis is the percent of control of activity for CYP2C8. And this is plotted as a function of increasing concentration of drugs. So on the right-hand side is gemfibrozole. On the left-hand side is gemfibrozole glucuronide, one of its major metabolites. Now in each of these plots, there's two different treatments. The black squares represent the 0-minute pre-incubation. So the enzyme is incubated with gemfibrozole or its glucuronide metabolite, and the marker probe substrate is added to it and the reaction is started right away. Or the open circles represent the 30-minute pre-incubation, where gemfibrozole or gemfibrozole glucuronide is incubated with the test system for 30 minutes prior to addition of the positive control probe substrate. And then, of course, the reaction is conducted and the amount of activity of the enzyme is measured. Now, if you look at the left-hand side, gemfibrozole, you see that the 0-minute IC50 and the 30-minute IC50 are pretty much the same. So we have 120 micromolar IC50 for 0 minutes and 150 micromolar IC50 for the 30-minute pre-incubation. However, on the right-hand side, with the gemfibrozole glucuronide metabolite, we see that there's a very strong leftward shift in the IC50 curves between the 0-minute pre-incubation and the 30-minute pre-incubation, where with the 0-minute pre-incubation, the IC50 is 24 micromolar, while at the 30-minute pre-incubation, the IC50 decreases all the way down to 1.8 micromolar. This leftward shift seen in the IC50 curve between a 0-minute pre-incubation and a 30-minute pre-incubation is indicative of a time-dependent inhibition of the enzyme. And in fact, fatal interactions occurred with cevirostatin, which is a substrate of CYP2C8. So if taking cevirostatin with gemfibrozole, which can be metabolized into this CYP2C8 glucuronide metabolite, can lead to an accumulation of cevirostatin, which could potentially be toxic. And in this case, gemfibrozole was withdrawn from the market in 2001. So this would be a good spot to point out that when investigating potential drug-drug interactions, it's important to also have a proper study design and use the right test system. And look at the totality of the data from both in vitro and in vivo studies. So some good examples of this are potentially opposite effects of glucuronidation that can occur, and interpretation of the results between different test systems, and how this can lead to different conclusions. So again, these examples, one of them is with gemfibrozole and the other ezetimibe. So gemfibrozole, again, which I just discussed in the last slide, has an acylglucuronide metabolite that's a time-dependent inhibitor of CYP2C8. And this is observed clinically. However, this inhibition is not observed in vitro with gemfibrozole when it's incubated in human liver microsomes and only considering CYP metabolism. So to state this another way, incubating gemfibrozole in human liver microsomes fortified with NADPH alone would provide a false negative result. So if only phase 1 metabolism is being investigated, in this case, a potentially relevant clinical drug-drug interaction could be missed. On the other hand, the opposite can be observed in the case of ezetimibe. So significant time-dependent inhibition of CYP3A4 is observed when ezetimibe is incubated in human liver microsomes. However, ezetimibe does not cause any clinically significant inhibition of CYP3A4 in vivo. So in this case, a false positive result is obtained with the in vitro experiment in human liver microsomes. Now, the reason behind this is because the glucuronide metabolite of ezetimibe is not an inhibitor of CYP3A4. And when ezetimibe is metabolized in vivo, it is rapidly glucuronidated. So therefore, there is very little of the ezetimibe present to inhibit CYP3A4. So the rapid glucuronidation in vivo is kind of protecting from the inhibition of CYP3A4. So just to summarize, the design of the in vitro experiment, the choice of the test system, and looking at in vitro and in vivo data combined is the best way to understand the true drug-drug interaction potential of the compound. So as these examples show, with glucuronidation, opposite results, time-dependent inhibition, protection from inhibition can occur due to glucuronidation. So in July of 2021, the FDA position on UGTs and drug interaction studies was presented at ISSX in a short course, and that's referenced here on this slide. So in the current guidance, there was little mentioning of the UGTs. So the main question that does need to be answered, is the investigational drug a substrate of these metabolizing enzymes? So the guidance states that first, the major CYPs need to be investigated. And this needs to be followed up by the minor CYPs and any other hydrolyzing enzymes that are involved in the clearance of the drug. If no sufficient answer is found there, then additional phase 2 enzymes should be considered. And these include the UGTs and sulfonotransferases, along with UDG glucuronosyltransferases. So some general things to take into consideration when evaluating the victim or perpetrator potential of a drug to UGT or to any other non-CYP enzymes can be put forth in the form of the following questions that the drug developer should aim to answer. These are UGTs the main route of elimination for the drug? Are one or more of the UGTs, and again, or other non-CYP enzymes involved? And are these expressed polymorphically? What is the likelihood of the drug being co-administered with other non-CYP inhibitors? Are there any active metabolites that are formed? And lastly, are any of the metabolites that are formed reactive metabolites? Now again, while these questions that the drug developer should aim to answer, while it's put forth with a focus on UGT enzymes, these same principles should be applied to other non-CYP metabolizing enzymes as well. So how do we address the reaction phenotyping of non-CYP enzymes? I'm going to illustrate this example again using UGTs, but keep in mind that this approach can be applied to other enzymes. So UGT reaction phenotyping can be conducted in a process that's similar to the approach that's adopted for the CYP enzymes. So based on the current FDA guidance, it's recommended that data from multiple approaches are taken together whenever possible. For example, when investigating the role of CYP enzymes in the metabolism of a drug, these experiments are conducted in recombinant CYP enzymes to assess which CYPs have the potential to be involved in the metabolism of the drug. Additionally, experiments are conducted in human liver microsomes with and without the presence of specific chemical CYP inhibitors to assess which CYPs are involved in the metabolism of the drug. And this approach should be followed as closely as possible when investigating non-CYP enzymes as well. So some key takeaways that I want to come out of this webinar today. First, CYP-mediated metabolism of small molecule drug candidates will likely dominate for the foreseeable future. As we saw with some of those aggregate studies at the beginning of the talk, 50 to slightly greater than 50% of FDA-approved drugs between 2005 and 2016 are still metabolized by CYP enzymes. Non-CYP metabolism is important, and it can be dominant for some drugs. So drug developers need a strategy to approach reaction phenotyping. And this approach can be based on structure, NADPH dependence, inhibition by 180 ABT, etc. Now, based on initial results, the approach taken to further elucidate the metabolic pathways of the drug in question, they might be very straightforward. As we saw in those flow charts, you might quickly come to the conclusion that FMOs or carboxylesterases are responsible for the metabolism, and the investigation ends there. However, you may need to take a broad approach with many enzymes being assessed, where you utilize many different test systems and the presence and absence of many different cofactors to elucidate which enzymes are involved. And more specific enzymes might require a literature review and a more in-depth in cerebro assay development. So there's not always a silver bullet or an easy pathway to take. Now, I'd like to just do some quick acknowledgements. I'd like to acknowledge all the scientists whose data was referenced on these slides and used in this presentation. I'd like to give special thanks to the XenoTech consulting team, Drs. Brian Ogilvie, Pallavi Limme, and Mashi Zerwinski for helping to prepare this presentation. And I'm also happy to acknowledge all past and present Sekisui XenoTech scientists who contributed to this field. I'd like to thank you all very much for taking the time to attend today, and I'd be happy to open this up to any questions you may have. So, thank you.